Wonderful. Okay. So, welcome everyone. We are very happy to have our seminars back, and today we're going to have two exciting pollination talks that will each last for about 30 minutes, including questions. And our first speaker is Coco Deng. And Coco, we have actually never met, but Coco reached out to me about a year and a half ago asking about vertebrate pollination in the Caribbean islands for a potential student project. And then she went on to the field to Montserrat Island and actually developed a very cool pollination project for not only a Melastoma species, but also a Campanulaceae species. But she's going to talk about the Melastoma Miconia species that I and I think many other people have been dying to know the pollinators of. Yeah, Coco and I share some interests, and that includes that both of us had wanted to combine our passion for birds and our passion for plants, and it looks like Coco managed really well with this project. And she also just recently graduated from Bard College at Simmons Rock in Massachusetts. So congratulations to graduating, and we're very excited for your talk, and you can go ahead and share your screen, please.
Thank you for that great introduction. Give me a minute to figure out the screen share. There we go. Okay. All right. So, hi everyone. Before I get started, I want to give a little bit of context on my research. So, I did this research for undergraduate thesis with my college's program for undergraduate research on the Caribbean island of Montserrat. This was a self-designed research project, so I'm very excited to share my work with everyone here today. So, a bit more context on like why I did the study. So the goal is to learn more about how native and endemic vertebrate pollinators interact with a plant endemic to an area of the Caribbean. And though this is a small study, this is the first study on the pollination of Miconia purpurea. And Montserrat itself is also an understudied but really, really biodiverse island. And like a lot of other small tropical island ecosystems, and the endemic species that live within the Montserrat is really vulnerable to man-made changes like climate change and human development. So it's really important to understand the ecological relationships of the flora and fauna. And a bit of information on Miconia purpurea. It is endemic to the Lesser Antilles region. In Montserrat, we found it growing in cloud forest and wet forest of elevation about like 600 meters or above, which is for Montserrat, that's pretty high elevation. We found it flowering in January when we did our study, as well as in May when we returned for a second visit for my lobelia research. Um, I've also found that it's really attractive to nectivorous birds. The fun thing is that you can kind of hear these trees before you see them because you just hear a lot of banana quits and hummingbirds kind of calling and being territorial before you see the trees themselves. And cloud forest and wet forest is also threatened, one of many tropical habitats that are threatened because they require constant humidity and a lot of rainfall. So it's really sensitive to any shifts toward a drier climate, among other things. I should say that the conservation status of Miconia purpurea hasn't really been assessed, but it is growing in, has a very limited range and it's growing in a threatened environment. Um, so for this study, I want to focus on vertebrate pollination because I was interested in how birds use nectar resources, but based on many things, we also hypothesized that vertebrate pollination would be kind of the main form of pollination for Miconia purpurea. It's got a very kind of like classic generalist bird pollination syndrome with a short corolla that's bright red. It doesn't produce a scent and produces a nectar reward. It was also formerly in the Lesser Antillean endemic genus Carianthus, which has been known to be visited by hummingbirds. And my advisor, Donald McClellan, had seen a banana quit visit one of these plants before in Montserrat. So we also thought that based on the anthropomorphology, buzzpop insects are a bit unlikely to contribute to pollination, even though we have seen butterflies and flies kind of buzzing around the flowers and probably probably getting some sort of reward. Um, I've talked to Agnes a little bit about this, and this is, correct me if I'm wrong about this context. But so, as many of you may know, buzz pollination um, is likely to be the ancestral pollination habit of Melastomataceae, from which vertebrate pollination is derived. And so for Miconia that's functionally specialized for buzz pollination, you get poricidal anthers that are kind of like what you'd see in Solanaceae with narrow slits that are where pollen's released by buzzing. And then with vertebrate pollination, you get these sort of, I don't know if you can see my mouse, but if you look at the pictures, you get these poricidal anthers still, but they've been adapted to have much wider slits. And the anthers themselves are more flexible, with the pollen being released by this salt shaker mechanism, to use Agnes's terminology, with the pressure of a bird's beak or a mammal's tongue releasing the pollen from these wide anthers instead of buzzing as you would see in insects. Because Montserrat is such a small island and it's pretty understudied, I want to give a little bit of context on that. So, island itself is a British overseas territory with a total land area of 102 square kilometers, and the highest point on the island is just over 1,000 meters at the summit of the volcano. So, the Lesser Antilles is this area of the Caribbean where you see this arc of small islands. And that's been produced by the subduction of North American tectonic plate under the Caribbean tectonic plate. And that produces a lot of volcanic activity and seismic activity. In Montserrat, you get several mountains that have all been created by volcanic activity. And from the north end of the island to the south end of the island, you get these smaller, more eroded mountains. And then you get up to the Center Hills Forest Reserve, which is where my two research sites were. Um, and then at the very south end, you have the Soufriere Hills, which is still volcanically active and produces volcanic disturbance. And because the Soufriere Hills is active, the Center Hills represents the oldest and most intact moist forest habitats on the island. And I did this research from during just a short period in January. I was hoping to do some more stuff in May, but the flowering period didn't quite work out. So I've mentioned the volcanic activity, but Montserrat is subject to a lot of frequent human and natural disturbance. Colonialism introduced a lot of invasive flora and fauna to Montserrat over over the years. So that includes organisms like guava and cane toads and invasive rats that can be found even up in like higher elevation forests where we were working. Montserrat is also in the hurricane belt. And volcanism really does have a huge effect on this island, especially given its small size. The Soufriere Hills volcano produced several eruptions from 1995 to 1999 that covered roughly a third of the island in volcanic ash, and really heavily affected the ecosystem. So you can see here, this is a picture of the Center Hills where we were working, and to the right is a picture of the Soufriere Hills today, and that would have looked like the picture to the left at one point pre-eruption. So a lot of ash ran down these valleys and may have caused the extinction of one of Montserrat's three known endemic plants, which was a small Salicaceae shrub. Um, so cloud forests are a bit lucky in this regard because their higher elevation protects them from being covered by ash flow as well as human development agriculturally. So I just wanted to emphasize that while this study focuses on vertebrate pollination, we didn't like really critically empirically look at the effectiveness of invertebrates, and that's because vertebrates was our focus. So my research questions were simply what vertebrate pollinators visited Miconia purpurea? Does the pollinator and plant morphology match? That is, can birds comfortably access the nectar? And are trail cameras effective for observing vertical pollination? There's been increasing research on using camera monitoring for pollination, but there still isn't a lot of research on this. So we were curious about whether it would work in this context, because trail cameras are a pretty cheap way of getting a lot of observation hours and reviewable footage. So to answer two of my questions, we observed pollination in the field, and the way I quantified it was I kind of defined one visitation as when I saw an individual insert its mouth parts into the corolla, to when it disappears from view or just stops feeding. So with in-person observation, I spent a little bit of time at one site, and with camera trapping, I set them up for a bit longer at both study sites. You can see here that's Donald right there. But up here in that red circle, that's where we had our camera. Miconia purpurea trees grow up to maybe like 3 to 5 meters-ish. So we mounted our cameras on these poles so that they could be like closer to the flowers. Trail cameras themselves, we got this commercially available Browning camera that's relatively cheap. I selected it because it has a faster trigger speed, which would be nice for capturing really small, fast-moving organisms. It also has, well, it's been advertised to have invisible infrared at night to avoid scaring any nighttime pollinators potentially. And then I set it to take 4 photos per trigger. So whenever it perceives movement, it'll take 4 rapid shot photos and then a small break, and then more photos if it detects more movement. With morphology, I was just trying to figure out if the bird's bill lengths were sufficiently long enough to access the nectar reward without having to resort to like robbing. So I compared the corolla length and culmen lengths from literature and databases for birds, and I got corolla length from digitized herbarium specimens that I measured in Photoshop by just tracing a line along where the scanned specimen includes like a scale, and just using that scale as a ruler to like measure the corollas. That makes sense. Not a lot of collectors come to Montserrat. So for a lot of my pollinators that have ranges beyond the Lesser Antilles, I made the best effort to obtain specimens from within the Lesser Antilles range. All right, on to my results. So what vertipollinators visit Miconia purpurea? Um, we So a lot of bird visitation. It seems like the main vertebrates I visited were birds. So there was a significant difference between how often different pollinators visited. The purple-throated carob, which is a large hummingbird, visited the most times, with banana quits and Antillean crested hummingbirds also being pretty frequent visitors. And I'll go into the pollinators in a bit more length in the next few slides. Um, we also we also had a rat visit a couple times, and we did see when we I reviewed the trail camera footage, I did see a bat in frame, but it was pretty low quality, and I couldn't determine whether it was a legitimate pollinator. There are 3 species of nectivorous bats on Montserrat, so it's definitely a possibility I wouldn't rule out. I also spoke with Julia De Janeiro, who is a PhD student in Nathan Muchala's lab at the University of Missouri St. Louis, and her lab works on campanulaceae and bat pollination. And she was saying that for nighttime observations of bats, sometimes trail cameras and the trigger speeds won't be fast enough to capture it. So that could also be a reason why we didn't get any bats. Something that was kind of interesting I noticed was that during the short time I was down there in Montserrat, we there seemed to be a difference between what pollinators visited when it was raining versus what pollinators visited when it was clear. Um, so you can see here when it was raining, we got a lot of these purple-throated carobs, as opposed to when it was clear, we got a lot more banana quits and Lesser Antillean or Antillean crested hummingbirds that are more generalist, and with orioles and rats, we didn't really get that many in general, so there wasn't a huge difference. So with the pollinators, we did get the purple-throated carob is a Lesser Antilles endemic as well. It's one of the primary pollinators of two heliconia plants, actually, with the females feeding on Heliconia bihi with longer corollas, which is why they have a longer bill, and the males, which are slightly larger with a shorter bill, feeding on Heliconia carabea. In addition to females, they are sexually dimorphic, and they've been known to be pretty aggressively territorial with their nectar, which is something we saw with the Miconias as well. They were always kind of fighting amongst themselves and other birds. We also got the Antillean crested hummingbird that's also endemic to the Lesser Antilles. It's smaller and more generalist than the purple-throated carob, and it's found in a lot of habitats in Montserrat. You'll see them in dry and mesic habitats often as well. It's a much shorter, straight bill. We got bananaquits, and those are generalist nectivores and frugivores that have a lot of variation throughout their range that extends to South America. Um, So when we were looking at specimens, we got specimens from the Bartholemica group. That's the Lesser Antillean kind of group slash subspecies slash taxa. And they're also found in a lot of habitats in Montserrat, including like more lowland habitats and human developed habitats. Something that we were considering was that the avian visitors to Miconia purpurea might also reflect the tendency for island pollination systems to be a bit more generalist. And Montserrat is a really small island, so generalists like Antillean crested hummingbird and bananaquits, which are normally found at lower elevations, so not in like highish elevation forest, may be forced to move up higher in elevation to find more resources. Something that was really exciting was we got a few visits from the Montserrat oriole, which is endemic only to Montserrat. This bird is known mostly as an insectivore. It feeds on some fruits, but there hasn't really been activity reported for this species. This bird is found in mesic to wet forest. And although we only got a few visits, it was really consistently seen at one of our study sites and was also seen to be quite territorial, especially when it rained, which was interesting. Um, this is exciting because the Montserrat oil population got pretty heavily hit by the volcanic eruptions, and though they have recovered quite significantly due to conservation efforts, they are still a very small population and are vulnerable. Without more observations, it's tough to say how important Miconia purpurea is to the diet of the species, but it could be really important to note that Miconia purpurea is one of the plants that the oriole depends on. One of our surprise visitors was the introduced black rat. This was introduced to Montserrat by colonization, where it is now pretty widespread and invasive. It's been known to prey on native fauna, like some endemic amphibians and some endemic reptiles, as well as eggs of native birds in forests. And so Montserrat doesn't have any native mammals originally, except for bats. So we saw this visiting only a few times, and I just wanted to note that to get to to get to the Miconia purpurea flower, it would have to climb about like 3 meters up the tree, in addition to being at an elevation of almost 600 meters, showing that it's pretty willing to get to food resources and also disperse into these forest habitats. So with pollinator morphology, I measured a few Miconia purpurea corollas and as well as collecting a bunch of measurements of bird beaks from literature. So this was both from I gathered some averages as well as looking through databases like GBIF to get Coleman measurements. And as you can see, this green line here is the length of the Miconia purpurea corolla. And then you'll see all of these birds' Coleman lengths are above that threshold. A lot of the pollinators have longer Colemans in the corolla, which would imply that they can pretty comfortably access the nectar. so that even the smaller generalists have longer culmins than the corolla. So the short answer to whether trail cameras were effective in this case was, well, not quite. Um, so the pros of using trail cameras for this study was it made nighttime observation possible and also safe. Our study sites were pretty high up in forests. You had to use pretty steep trails to get up to them, so observing at night would have been not only difficult but really unsafe. And we managed to get a lot of observation hours and reviewable footage without causing disturbance to pollinators, especially pollinators like rats potentially that would be spooked by people. But we also got very few observations compared to our in-person hours. We only spent a few hours in person at our sites, but the in-person hours yielded a lot more observations. So part of that reason could just be we to get trail camera observations, you need to leave them up for a bit longer. Um, Usually with longer term studies, you'd be able to do that. As I kind of mentioned before, trigger speed might also be a limiting factor where you get a lot of small, very fast moving vertebrate pollinators coming in and out. So the camera might trigger, but it triggers too late and it misses the pollination event. Um, Cameras have been successfully used before for both vertebrate pollination as well as bee pollination, so it definitely shouldn't be dismissed as a good tool. But in this study, it seems that in-person observation was a lot more effective. So to conclude, I did identify the vertebrate pollinators of this plant, although more observation hours and greater sample size would make that more solid. And there's also more methods that could be used to give us a fuller picture of the pollination of this species. One future direction that I'm that I would like to try is manipulative experiments like exclusion experiments, so bagging the flowers in mesh. That would allow us to definitively determine whether insects contribute to the pollination of the species and how much, if so. Pollination rate is another item that you could get with more observation hours, as well as nectar analysis, which you could do if you had access to the equipment and expertise. All right, that's the end of my presentation. Thank you for your attention thus far.
Thank you, Coco. Wonderful. That was really fun to see all of it. Very cool. Well, part of it, since you also worked on the lobelia. Are there questions from anyone?
Of course, I have a couple. So very quickly, Coco, you mentioned that for the hummingbird, there's dimorphism between in the beaks of male and females. Did you notice a difference in, they were your most common visitor, but were you had more or less same amount of males and females, or was it different amounts of males and females? And the other thing for the banana quid, and a lot of things when they are robbing nectar, they go through the side of the flower rather than through the top of the flower. So if they're going through the side, they're really robbing, not they cannot be potential pollinators. What did you observe on that?
So with the purple-throated caribs, I think they were just like moving. They would move in and out really fast, and that was one of the metrics I tried to get, tried to observe. But it was just really difficult to get tell male and female apart in the space of like a second when it comes in and it comes out. With banana quits, I didn't notice it going in from the side, like putting its beak in from the side. Although I definitely wouldn't dismiss it. It's definitely like a really, really generalist bird.
Is there another question from someone in the audience?
I have a question.
Hi, Winnie.
I hi. Thank you, Coco. A very nice talk. I just wondered if you saw some bees also on these flowers, and if not, what do you think is excluding the bees from these flowers?
I think I was not, I wasn't focusing on the vertebrate pollinators as much when I was at the sites. I did see flies and hoverflies, and I feel like with the amount of attention I gave them, there could have definitely been bees there. If there's any factor that's like excluding the bees, I don't potentially elevation. I know there's been literature on like elevation being a limiting factor for some vertebrate pollinators, but or invertebrate pollinators, but then again, my study sites weren't really like as far up as some of the ones in the literature. So yeah, I don't know if there would be like a lot of barriers to vertebrates feeding on these flowers as
So the
well.
red color is not really a bee color. That particular purplish red.
Also, the stamen, I'm wondering whether bees or small insects could pollinate because the stamens are so far exerted. So even if they can get to the nectar, they probably don't really get the pollen on their bodies.
Exactly. It's not suited for buzzing.
Mm. I have a crazy question, Coco. So if introduced rats, just common rats, visit the flowers, should then not rats also in North America and Europe and just the temperate zone in general visit flowers? This is a crazy find of yours.
Well, I guess I haven't considered that, but then again, I haven't seen any literature of someone putting up cameras in North America to see if that happens. I do
Yeah,
know that one study I found when I was looking for like studies that looked at vertebrate pollination and trail cameras, where it was, I think, Australian proteaceae, which is partially rat pollinated. So there is that. But I think that was actually native rats and not the European black rat.
yeah. Proteaceae are a crazy group. I think they also have rodent pollination in the Andes, so it makes sense that they are doing something wacky again. But it's just if a rat can adapt to nectar flowers, why don't they just maybe, as you say, maybe it's a lack of us not looking at night in the temperate zone. Maybe there are rats everywhere going to flowers. Does anyone else have a question or a comment or a position to offer to Coco for her PhD?
I do have a question because Caribbean Melastomataceae, are there any coliferous, cauliflorous Myconias on Montserrat?
Um, hmm.
The reason I'm asking is that somebody sent me a photo of what he thinks is a Miconia, but it's coliferous and it's from an island nearby. What's the island? It's an island nearby. It's not Montserrat. And it looks very similar, except it's cauliflowers.
Oh, that wouldn't
Okay,
that might
okay.
be a better question for Agnes and for me, but that is really cool.
Maybe it's
Fabian might know which species that is.
yeah. I
Yeah,
will get
there are a couple of Henrietteas in the Caribbean.
not a Henrietta. I will send you the photo. I will send it. It really looks like purpurea, except it's cauliflowers. So I will send
Yeah.
you the.
That's
Never
Yeah,
crazy.
do send me the picture.
Maybe it starts
Um,
adapting to rodent pollination.
so I will
Um,
stop this recording
mhm.
and then start the next one
Yep.
for